Good morning, good afternoon or evening perhaps, or you might be joining us. This is Divestream 870 of the remotely operated vehicle Sebastian. We've just come down a little less than 200 meters from the surface of the ocean, and currently we are looking at some corals here. We shall see what the push core game is today. We're not planning on this being a particularly long dive. It is quite shallow. We're actually hoping to squeeze in a second dive today. But I'm going to bring up the microphones for the rest of mission control, and you'll be hearing the chatter going on here as we dive 200 meters below.
Whenever he is, I just got him shuffled off 50 meters, but just basically he is. Whenever you need, where the pool is. What is just wait.
Oh yeah.
Just wait, pal. Just hold on. The white, some of the white corally things, whenever you have a chance to.
It does look softer than the other ones.
So Brett, above and to the left, there's white that's more stringy and flowing that we're more interested in than the coral.
Whoa!
Yeah.
To the left?
Yeah, to the left. Yeah.
Stringy stuff?
Yeah, white. Yeah,
Oh.
right there.
That one.
Yeah.
That looks like sulfur filaments.
Yeah, those look
Yeah.
like veggie.
Those are sulfur. Oh.
So it could be
Yeah.
Yeah.
Yeah,
yeah.
100%.
Oh, yeah.
That's
this looks like
like
sulfur. There's
Yeah,
like
it looks like
sulfite.
Veggiatoa,
Yeah.
She's what?
sulfur oxidizing.
Oh, I see. Okay.
Sweet.
Yeah, they took filaments
Yeah.
of sulfur. Yeah.
Awesome.
That's the.
Wow.
That's
So
what we want.
it's bacterial.
Yeah.
Yeah.
Yeah. Stick a fish core in there.
Could you
We
suction
could suction
sample
it. We
it?
could suction it. Yeah.
Scoop it.
That would
If
be
you could-
wonderful.
If you could suction that, yeah, that'd be great.
Yeah.
Do you want to explore around or just?
Yeah, that bit or.
Yeah.
Get it.
Definitely get that bit.
I think before exploring, let's get that one.
There's more right on
There
the
is more?
bottom right. Yeah, I saw it when we were looking at the coral.
Sweet.
That bit's good.
Yeah, that's a big chunk of it. Beautiful. Big old fish.
That's flow. That's subsurface flow.
There has to be. Can we get a sense of the temperature? Like
Yeah, it's 8 degrees roughly.
8?
Yeah,
8?
It's right here. Yeah.
I mean, we can stick the high temp in there, but it doesn't.
I mean, we're
there's
only
any shiver,
200
so.
meters, so
Yeah,
that's
but
about
still,
right.
like it's very high temperature.
That's pretty consistent with what we've
With
gotten
this
on the
gas,
CTD.
yeah.
Yeah. It's actually cooler than the other spot we're at.
At 200?
Yeah.
Yep. You got that Brazilian spicy water.
It's not minty yet. Is what you're saying.
No. It's well, that's not super salty, so I think it's maybe a
Did
transition.
the temperature go up when I got closer? Has anybody been watching?
No, it's been 80.
No, it's been
It's been 8?
consistent.
Okay.
Yeah, the whole time.
So it's not.
Nothing crazy.
Yeah, that would be really.
Yeah.
Yo.
What's it called again? I think.
The
What's it called?
white
Yeah.
that we're suction sampling, it's Veggiatoa, so it's a
So
sulfur
those are like
oxidizing,
when you
usually
look at the,
bacteria.
Like, the real ones,
Yeah.
you have like a top
Yeah.
white filament with different colors. This one is white, but they can be pink, they can be yellow, they can be
Oh,
orange.
I was just, yeah, so I could see I was just moving
But
it.
it's
Yeah,
like
this is
to-
fine.
It's like filaments.
Yeah,
Yeah.
absolutely.
R1.
Yeah, so
That's
those
cool.
white strings there that are flowing.
Awesome.
Yeah. That is super cool. I was not expecting the Yatoa here. So that probably there's a sulfur source. Because where is it? It's like oxidizing sulfur. So it has to have a sulfur source in there. Yeah. I'm wondering if it's
vacuuming the seafloor.
Yes.
Come on, little guys.
Beautifully done.
Oh,
Wow.
beautiful.
It's really soft. Though.
Yeah.
Yeah.
There they
Yeah,
go.
that's
Yeah.
good.
Been in there.
Good.
Wow.
So,
Yeah.
Yeah, that's
Okay.
good. Yeah.
Brilliant.
Wow.
Thank you.
trying to see what were the differences in Vegetula's color because we don't know why they
Just stay here
have
for a second.
different
Let's just see the-
colors. They
keep
have
zoomed
different
in on it for a
colors
second.
depending on the temperature.
For biofilms,
And they
yeah.
can be white, they can be yellow, they can be orange.
Brett, what's our mesh size?
You know which mesh was on that?
Just with the number.
Just number one.
Just number one.
Sorry, there's a selection for small, medium, large
Oh, it's
mesh
medium.
size. Thank you.
We can do it in different
Oh,
jars.
awesome. Yeah. Just maybe try and get a little more in
In a different jar?
Yeah.
Which jar would you like?
What's what we can get to? I guess what is.
We can.
Oh, another medium.
We can index around. Not sure what the.
Oh yeah. Let's see.
5, 6, 7, 8. I think. What have you got? Medium or large?
Yes. Medium.
Medium is one is medium already.
Okay. Okay.
So the deep
And
colors.
8, you want some.
Yeah, let's try.
I'm not sure how deep they go. Quite a bit deeper than this, though, I think.
Cool. Thanks.
8's on.
Or you gotta respond to
Oh.
That's good.
Good.
Yep. Cool.
Yeah, so.
Yeah, sure. Right. Let's explore. That's great.
You want to just skirt around the outside of this mound and see what we see.
That sounds good, yeah.
That's great.
Yeah. Is the mic's on?
Yes.
Let's try.
What's that?
Yes.
Hey everyone, we're just pretty busy. We're distracted. We found this about 2,000 meters. We found on the multi-beam last night. These mounds. So we dove. We went, and now we just got down, and we're seeing these white hairs, and we're seeing on these mounds. You see these little areas where there's like streamy things of white hair. That's bacteria. There's like a few different types of bacteria that do this, but their common one is Beggiatoa. There's also like Thiothrix. These are bacteria that breathe hydrogen sulfide gas from the subsurface. So we think that we know that there's sulfide flowing up from underneath this mound so now we're just exploring around the mound a little bit.
because we're going that way anyway, but keep them like above us. Yeah, yeah. But maybe a little bit further north, so we don't get close to the reds.
Cool. Yeah.
Wow.
Zoom in on that.
It's quite strong currents here.
Very, you have a new waypoint view.
Unfortunately, I'm getting more into the current.
Thank you.
Give it a good shot.
Oh. Just against that.
Yeah.
It's very soft.
Yeah.
That fish.
Cool.
Yeah, really cool.
There might be some bits we can take information and go down the ball.
Yeah. Let's look around a little bit first and maybe, yeah, might be worth trying.
So we're going
Yeah.
What? How extends this like area is? Like how many?
It's pretty wide.
Oh shit.
$1,500.
1,500.
1,500 across area?
Yeah.
Yep. Oh, that way, yeah.
Well, if that's 600, this is.
Oh, okay. Yep. Wow, it's huge.
It's huge.
So I think they'll go up to
Bump up the ISO one. Let's add more to the waypoint.
I have no idea. Like whatever's easy for you to like explore with and not kick up stuff. I think any direction from where we are, 'cause there's something on top of this thing.
Do you want to drop a marker here? We get up to this bit again. So wait till I'm actually on the land again. Drop a pin like in the middle of
on the slope.
Oh wow.
Yeah. On the backside.
I suppose we can't go down. Let's see what this is. What were you gonna say, Val?
No, I was just thinking if there's been like sulfur reduction and lots of conditions in the morning. Because we have kind of spores and stuff exposed, but mostly dead. It's not as like it's not as exposed. So I don't know if there's some sulfur coral.
Do some spread.
Looks pretty healthy when we zoom in.
Yeah.
INS position. INS is 3. INS position.
Associated with sulfur coral or not.
It's gonna add for weight on one. See.
Fish on fish. Lizard looking thing.
Yeah. Oh yeah.
You can go on there. We see that is white.
There's
Yeah,
another.
but the little pump, they're like shrunken. So we haven't used it. And then there's obviously the big solitary polyps that are corals too. Yeah, they're awesome. But none of these are in there.
Yeah.
Sulfide oxides with mats, there's none of our analytes or anything like that. That would be fun. It's probably pretty ephemeral.
Yeah.
They haven't, I don't.
Oh, did we stick a temp probe in those?
We did not the high temp. We should do the next one, but there's no shimmer, so
It's true.
I mean, like, you should.
It's pretty loose and soft.
Yeah.
I mean, you might even get up to like 15%.
Yeah, that'd be cool. Do you think? Yeah. Do you think it's groundwater intrusion?
High temp probe?
Yeah. Stick.
Yeah. Okay. When we see another one.
I don't think it'll be high temp.
No,
but
I
it
know.
might be a
Yeah, I
difference.
know.
Do you think like? Yeah. Do you think it's pushing groundwater intrusion from the shell?
I asked Allison. I mean, she just there's sulfur.
So if we see on the left
I mean, yeah. If we had a salinity probe, we could just stick a stick.
Yeah.
To top it.
Good ears. Good ears. Good ears.
Yeah, exactly. Like we have in California. I mean, very different shell systems. But yeah, could also be a fish pond.
There's little white patches, I think, are cool. I was looking at the polymer phone.
Yeah.
But
Yeah.
if you get a really close zoom on the pink ones, you can look for clue tips.
Here, why don't you come over here and talk in the microphone? Because yeah. If you don't mind.
This place, you know?
Yeah.
Or you can chair or bring the microphone
Oh,
over
no,
here.
you're good. Yeah. You're photogenic too.
You're photogenic.
Well, they don't have to know that.
That's pretty.
Yeah. So we dove on this feature that we saw in the sonar that Brett and Natalie spotted this morning. I think the intention is not necessarily to core here. Is that correct?
We might try. It looks like we might be able to. Right now, we're just trying to get a sense of things. Yeah.
My name is Jacob. I guess I'm one of the resident zoologists they brought along. Wow, I've never gotten to say that before. I think we're looking here at a soft coral or a coral relative, though my hunch is that the pinker, the darker pink anemone-like animals that we're looking at are true anemones. And one of the easiest ways to diagnose this is to zoom in really close on the tentacles and see if there are clubs on the tips. So corals and corallomorphs, which are essentially relatives of corals that have developed a more anemone-like soft-bodied lifestyle, they all have club tips. Anemones tend to have just tapered tips. Thanks for the zoom, Mike. Yeah. It can be hard to tell in situ. Our pilots are doing a great job of steadying up.
High current.
Yeah. High current and a lot of particulate organic stuff. Like when we dove the, like my experience diving vents, which is the only other time I've really worked on the bottom, there's not nearly so much to stir up. I kind of leave that to you all.
No, I'm good.
No, I'm pretty convinced these are anemones.
Mm-hmm.
Though,
Yeah.
the white polyps we're seeing are probably a type of coral. They're a couple different species, and then as Ian noted earlier, we see some solitary stony coral polyps, which are pretty cool. Did you already explain all about the sulfur bacteria, Brett?
I did, but you can do it again.
Yeah,
No, I
say-
can't.
say it again.
Well, so we've suctioned, I think, one patch. Is that right?
Two so
patches.
far.
Yeah, 2 patches of this white filamentous material, which we think is a giant sulfur bacterium. So these are, if it is what we think it is, these are bacteria that oxidize sulfur compounds that usually come out of the crust. There's some guessing here as to where this sulfur might come from, but typically in other settings, these are found. These kind of mats are found in sulfurous springs, near hydrothermal vents, etc.
Jacob, maybe a question for you from the chat. Does radial symmetry in any way benefit soft corals?
Does radial symmetry benefit soft corals? That's an interesting question. It's clearly clued into body plan and evolution of
Yeah.
cnidarians. They are in fact radially symmetrical. Unlike us, for instance, we're bilaterally symmetrical. We have a left and a right side. Anemones and their free-floating relatives, Medusae, we're looking at some right now, they're all radially symmetrical. I haven't read any papers that speak specifically to the advantages of being round. And I think it would be a challenging hypothesis to test scientifically, because all cnidarians, anemones, and jellies are more or less round. Some are kind of more oval than others. But I would encourage you to look it up. It's a really interesting question. Nothing's coming to mind for me, but it seems to have served them pretty well, because based on the fossil record, we think jellies have been around for well over 600 million years. Interesting question about corals here being bleached. So, actually, maybe we should let our resident symbiosis biologist speak to this.
Oh, brutal.
The microphone on your right, Ian.
Yeah, so we are sufficiently below the photic zone, especially here, that it's very likely that these are not symbiotic corals. So most of the corals that we know and love are much higher up in the water column, and so they're receiving lots and lots of sunlight, and so of course they have this iconic symbiosis with photosynthetic dinoflagellates. But what's really cool is that many corals actually don't. They rely exclusively on feeding with their polyps on heterotrophy for food. It is interesting though because there are some mesophotic reefs. So there are some reefs that go from roughly like 60 to maxing out about 150 meters or so that still rely on some level of photosynthesis from symbionts. And so basically once you get below about 150 meters, especially in somewhat turbid waters like these, there's just not enough photons, not enough sunlight that are hitting the bottom to actually support symbiosis. And so part of the reason that they're white is, of course, that they're lacking these symbionts. They just don't have another kind of pigment. So much of the color in corals that are near the surface that we know and love, that is driven by their photosynthetic symbionts. And so, of course, when they bleach, this is when we know, when they're losing these symbionts, the symbionts are sort of bailing out. And so basically, these guys probably just never had symbionts to start with. Because this is probably just enough below the photic zone that you're just not going to get enough light to support that kind of symbiosis. And they're probably just feeding on all this delicious particulate matter that's just hanging out in the water.
Does anybody know what the really cool butterfly-looking fish are called?
Yeah, we got some bellow fishes here. So the striped ones are probably centroscope.
Cool. Thanks, Alex.
Bellow fish at least.
So are we looking for another one of these mats?
Yeah.
Get that?
If we can find, yeah.
See if we can spot one. Did you waypoint them? The ones that you picked? Or marker them?
No. Just got the mound in general, but
Yeah.
Do you have the if we want to make a directed effort to return to them, we can get those coordinates off of Sea-Log from the suction collection?
we've got the snail trail. We've got a marker on that point there. See on
Oh,
the
yep.
snail trail on mine where
Never
it's
mind.
thick.
They have the technology.
Gotcha.
See a bit on about where we're stopped, obviously, and we've got a big, there. Correct, it's about there. Drop another pin there.
So Jacob, how many? Each small thing is an individual, or?
Yeah, in this case, each little flower-like animal is an individual. Doubles back a little bit on our
Looks like there
confusing
might be
discussion about individuals and siphonophores.
It looks like there might be more Veggiatoa there.
Where?
Above the stony coral center going towards the top.
You see one where?
I
Oh,
believe
yes,
so.
yes, center frame.
There you go. It's smaller.
Bingo.
just there.
Good eye, yeah.
Are you able to get in there, Mike?
What,
Doesn't-
sample this or?
I think we want to.
Just stick the high temp probe in there, yeah.
That almost looks shimmery. It's probably
What
just.
the hell was that?
That's the big, the
You
effect.
don't have to worry about the FCC here.
I don't care about them.
What happens to a microbiologist when they see a Veggiatoa?
Still works.
They drop their laptop.
Yeah, so corals, like siphonophores, can also be thought of, or thought of by most as colonial cnidarians. But if you look at a bunch of coral polyps in a colony, they're not really specialized. Whereas a siphonophore, or even some hydroids, which are the bottom-dwelling parallel of siphonophores, the individual zooids can be highly specialized. So some of them are only capable of eating or reproducing or swimming.
Another question in the chat related to sulfide oxidizing symbionts. So yes, the classic iconic Riftia tube worms that we see at hydrothermal vents, as well as their allies, do host sulfide oxidizing bacteria. They're a little bit different from if we're sort of positing that these here are Veggiatoa. And if so, that's a sort of different group of bacteria than most of our classic canonical symbionts. But they're basically doing the same kind of thing. So they're fixing carbon, they're primary producers, and the way that they are doing this is it's kind of like photosynthesis. So of course we're turning CO2 into sugars, but photosynthetic things like plants and cyanobacteria, they're using sunlight, photons from sunlight to do this. Whereas these bacteria are using chemosynthesis, and so they're exploiting energetically favorable chemical reactions. So basically when you take reduced sulfide, it really wants to give away electrons, and you have oxygen, which is really great at taking electrons. And when you combine the two of them, it releases energy. And so these bacteria are sitting in a really great spot where you have this really electron-rich sulfide. It's reduced chemically, and right at the top, of course, this water is quite a bit of oxygen. As you can see, we're looking at 73% saturation, like 270 micromolar. So they're combining the oxygen and the sulfide, using that to grow and sort of generate their own biological energy.
Okay, the pilots are setting up now to pick up the high temperature probe and see whether the water that's rich in electrons, presumably as Ian noted, that's coming out of the seafloor here, is any different in temperature from the seawater around.
Val, we're getting a few requests if you want to add a little bit in Spanish.
I can't see.
Oh. You want to come over here, Val? Open seat. La doctora de Anda va a traducir para nuestras radioescuchas hispanohablantes.
A 200 metros de profundidad encontramos
Val,
unos...
you're talking to yourself.
Stand by, she's going to open your mic.
Oh,
Your mic isn't open yet.
oh shit.
Well, you're on.
I was like, ooh.
Check with.
¿Ya?
You good? Yep.
Ok. Hola a todos, buenos días, gracias por estar aquí, gracias por sintonizarnos. Soy Valeria de Anda y estamos a 200 metros de profundidad. En el mapeo de anoche se encontraron unas estructuras que Chief Scientist Brett Baker decidió explorar y bueno, con la sorpresa muy grata de que encontramos corales y estamos explorando ahora lo que está haciendo el Sebastian es en ese brazo tenemos una estructura para medir la temperatura. Los filamentos blancos que ven ahí al fondo son lo que parece ser Vegiatoa, que son organismos que utilizan azufre reducido para poder crecer. No sabemos de dónde viene ese azufre, entonces por eso estamos tratando de ver si proviene de una fuente geotermal o proviene de otros organismos como pueden ser sulfatorreductores, que también pueden proporcionar el azufre reducido que necesitan estos organismos que utilizan el azufre para poder crecer y sobrevivir. Y es una sorpresa muy grata porque hasta ahora no habíamos visto estructuras rocosas en donde pudieran crecer estos corales. Y bueno, ahora como pueden ver ahí, esos son filamentos que parecen ser Vegiatoa. Y bueno, ha sido una mañana muy, muy emocionante. Así que la temperatura no proviene de una fuente geotermal, sigue siendo 8.5 grados centígrados, 8.6.
Makes more sense.
So it could be sulfur reducers.
What's the conclusion? It's a hydrogen sulfide leak, but it's not.
Well, water
Yeah.
doesn't need to be hot to
Yeah.
contain sulfide.
Yeah, yeah.
Yeah.
It could be.
Just gotta check, you know. Do our due diligence.
Would you like to?
Y bueno, esta estructura que encontramos va a 1500, o sea, abarca unos 1500 metros, o sea, es bastante grande, donde pueden haber corales y se extiende a unos 600 por 1500 metros. And that's a question for you, Jacob.
This place is in the last question there in English.
Yeah.
I'm curious what they mean by displacement, like by our vehicle, or yeah, I could use some clarification there. There has been some interesting work done into the resilience of at least anemones that live in tide pools, and it's been found that they are very sort of mechanically resilient. Granted, the environment of a tide pool is pretty different from the deep sea, but since these anemones at least don't create hard skeletons like corals do, there's a possibility that they can go back pretty quickly.
I said all of that into a closed microphone.
I was gonna say, I was like, oh, dang, you know.
Sorry, I'm holding the button down by instinct, by the sampling equipment. So I can't speak to the resilience of deep sea anemones. They probably grow a bit slower than those that you would find in, say, the tide pools, and they're also not evolved to live with the same kinds of mechanical stresses as tide pool anemones. But I am aware of some studies in tide pool anemones. There are actually some, I think, done at UC Santa Barbara where Allison works, where they basically kept people out of a certain area of tide pools and then had another treatment area where people just trod on the anemones with boots for weeks on end, and the anemones were basically fine. These ones are a little bit different, but since they don't make hard skeletons like hard corals, my hunch is that they should be able to, that they should one, be fairly resilient, and two, be able to regenerate fairly quickly. Something else that's physiologically interesting about anemones and all other jellies is that unlike us, they don't have clothed circulatory systems. So if you imagine, one of us getting a cut, you bleed and eventually it clots. For some other animals that don't have closed circulatory systems like snails, it does not clot, and that's a real problem. But for anemones and jellies, this isn't an issue at all because they have no blood. They just rely on water diffusing through their bodies to deliver oxygen to their tissue, and that gives them a lot of regenerative abilities that animals that we think of as more complex, like ourselves, don't have. Our pilots are also making a really solid effort to disturb the substrate as little as possible, and that's part of what the arms on Sebastian, I think, are for. They allow us to stabilize without parking the entire vehicle on the bottom. Correct me if that's off, Mike.
The rounds on the side. Yeah.
Yeah.
I suppose it's still the broader question of, as scientists, we often disturb the thing we love and want to find out about.
We've done 2 sections of this, so if there's another one, it'll be the third.
En transmisiones anteriores, algunos de ustedes preguntaban acerca de por qué no había corales. Habíamos hasta ahora visto todas las zonas que hemos explorado. Los sedimentos eran bastante blandos, pero esta sorpresa del mapeo que se realizó anoche, se pudieron identificar estas estructuras y con la sorpresa de que encontramos estos corales que se extienden, como mencionaba, por 1500 metros y estamos empezando a explorar esta área. Y seguimos en las costas de Uruguay a 200 metros de profundidad.
I should have taken a phone picture of it. Something really interesting that happens. This is reminding me of a spot that Ian and I actually dove on off of Los Angeles about a year ago. I think it was Thanksgiving, and it's like a warm spring off of Los Angeles with a bunch of these filamentous sulfide oxidizing bacteria, and the local snails that ordinarily eat kelp, or algae, were chowing down on the bacteria. And people have actually done some isotope studies of snails from that site, which would lead me to wonder, just like Ian was talking about, this chemoautotrophy, as we scientists with our fancy words like to call it, this sort of generation of chemical energy by mixing chemicals that come out of the crust, can form the basis of an ecosystem, just like harvesting energy from the sun can. And so I was wondering how much it could pitch into the total supply of food in this ecosystem. But it does seem like a lot of the food coming into this ecosystem probably comes in the form of plankton that's getting caught by the anemones and corals.
The fishing line?
Yeah.
Did we mark that, you want to mark that anthropogenic?
That's right. It's too dark and stuff. Appreciate it.
Not the best visibility.
Yeah, it's trying to balance having loads of lights on and
Yeah.
stuff that's in the water. So
Yeah.
trying to find the balance. I never want the lasers, but science wants the lasers.
Science? Do you guys want lasers?
I don't think we need them
Okay.
unless you have another idea.
Are you gonna drop them on the bottom? You just don't? You're just asking if we want them on or off? I don't think we need them unless we find another patch
Yeah.
of.
Just keep the.
Galen, do we think this is like how deep do we think this coral rubble goes?
Right at this site.
Yeah.
I'm not quite sure what the relief on the structure is. It'd be interesting to see how far down it goes. I would think.
Yeah, right.
I don't know if we can do that.
I don't think we have a long stick.
Yeah.
Longer than the temp probe.
Oh, right. I was thinking, yeah. Well, I'm not,
Oh, that's a nice one.
Yeah.
Yeah, let's probe that.
Yeah.
Yeah.
And also laser it, please.
Yeah.
Oh, there's a hole. This wall is.
Yeah.
This is a big hotch.
Yeah.
This
Yeah,
is
but
great.
it's-
We'll sell that here.
Some kind of vibe out. You know me.
Yeah, just land it and let it settle. Let it blow off. Current should clear it up.
It's awkward when it's trying to force you to fly straight into the hill.
Yeah.
Vamos a tratar de obtener una muestra de estos filamentos blancos, de estos microorganismos que hemos visto también en otros sitios, sobre todo son muy abundantes en ventilas hidrotermales, formando biofilms. Y ahora los vemos aquí en estos corales. Vamos a tratar de obtener
Beautiful.
un poco de estos.
The bacteria you can see with the naked eye.
Rare.
Como un recordatorio, hasta ahora la exploración ha sido, les hemos mencionado mucho acerca de microorganismos, de la importancia de que no los podemos ver, pero bueno, aquí hay un ejemplo, Brett nos acaba de recordar, que estas bacterias sí las podemos ver y podemos ver las estructuras que están formando. Hay otro tipo de microorganismos como tío Margarita,
This is what I'm trying to figure out.
son microorganismos que guardan glóbulos de azufre y
It's
estos microorganismos que parecen pelos blancos
Wormy,
los podemos ver a simple vista.
but not alive. Yeah, that's super thick.
So that's what I was saying, that it's like
Yeah, that
we've seen it in corals that look.
So
Like-
yeah, we're just talking about that. I don't know if it's coral or worm.
I'm
Yeah.
looking at it. Look. See if you look. I don't have the pointy thing, but that's interesting. That's really interesting. It's such a blue thing.
Well, that's
But I'm looking at the rings
veil.
of the openings in the tubes, and they have these little striations around them that
That are giving an area.
that are yeah.
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 It's giving.
What about the white veil stuff on the right?
That bluish stuff.
The bluish stuff is what is getting me.
Yeah, I don't know. It's weird.
Well, that wasn't even what I was thinking of, but it's also bluish.
Yeah.
Colors.
Yeah.
More sulfur oxidized.
See that blue patch on the to the right hand side of yeah.
Yeah.
And it has a different consistency.
Yeah, looks like more.
Yeah.
Yeah, more like grass.
Probably a fungi.
It's a good structure.
Well, it's not just anything chemically that can produce a blue color.
It's true.
You know, yeah. How would you feel about sectioning that? Quite a bit of this blue
Let's
stuff
do
going
that.
on.
Let's try the temp first.
The temp probe
Yeah.
first.
Let's go first.
Or I don't know. What do you think?
I think that's a fine idea.
Okay. Where do we want to temp? Is the question.
Well, why don't we do it in the middle of the giant patch?
Yeah,
The
yeah.
bigger the patch, probably
There's
the
some
more.
more. Good
Yeah.
eye. There you
That's
go.
nice.
Good eye,
Wow,
Mike.
it's like thick.
It may not be microbial.
Separate the suction.
Yeah.
Some suctioning on the more filamentous section and a separate one on the more groovy
I agree.
section.
Yeah.
Okay, so temp probe first.
Yep.
Okay.
On the big-
and then,
the big
and then we
patch.
watch Gabriella come over here and start down. She's on the bridge.
Oh yeah.
No, I did not. I didn't see any of that. You guys see it. She's
Es poco confuso si los tubos son de gusanos o de corales muertos. Hay triaciones sobre las bocas de los tubos y por eso no estoy cierto.
where's the mic? Mute.
I think so, right in the big
Yeah,
patch,
right in
please.
the yeah.
Oh,
Hello.
a swell shark.
Oh,
such a cutie.
cute. It's like no. It's like you're not gonna sample here. Excuse me.
What about eukaryotes?
Yeah,
Guardian of the reef.
yeah.
Excuse me. I'm gonna
This is a
sit
highly derived representative of the Asgards. Where I'm from in California, we would call it a swell shark. It looks like a very close relative. Must be a different species.
So beautiful.
Benthic shark.
People don't
Probably this one has a defense mechanism of being able to inflate itself if it's agitated.
Ooh.
That's why we call them swell.
Sweet.
He's
Probable que esta especie de tiburonito puede inflatarse si si es necesario. Inflarse. Sí.
We're going
Smack in the middle,
tiburoncín.
please. Perdón, Val. I'm sure you could have given a better explanation.
So yeah, it seems like
Can we see the probe temperature?
Can you pull up high temp?
Oh, I see. That's the probe.
yeah,
Wait,
it's
no,
not.
isn't that the seabird? That's background. Thank you. Well,
Not
that's our answer.
changing.
Can you get in any further?
Yeah, we can go further.
Ooh. Ooh.
It's very, very loose coral rubble.
They definitely look corals, like very soft, like they're not
Yep.
soft.
Not consolidated.
Yeah.
Yeah, if you look, if you
Yep. Okay, we won't then.
This is what I'm
That's fine. I don't want any samples here anyway.
Certainly we are not going to take.
Just tell her I don't want any samples
Well,
then.
could you please listen to the rest of the
Oh, okay.
sentence? One thing is that this is what I told her, that certainly we were not going to take samples there.
Yeah.
some are like sulfide resistant. But they, sulfide is it's a mitochondrial inhibitor.
Yeah.
So
Yeah,
so
yeah, yeah, I know.
yeah, who knows about the mechanism? Can't be sure
Yeah,
exactly
but I'm
whether it's
just
the
saying
anoxia or
like the
sulfide.
association between the places where we've been finding
Yeah.
the big Yatoa, it's in like
I agree with you.
exposed like dead coral, not in the areas where they look healthy. So I don't know if that might.
Okay.
Do you want to repeat your observation?
Oh, in the mic.
English or Spanish? We have the mic
Ah, en
back.
español.
Español o inglés. Es una observación interesante ecológica.
No sabemos si hay alguna, bueno, lo que estamos observando es la presencia de estos microorganismos que utilizan el azufre reducido, es H2S, y este azufre reducido puede provenir de diferentes fuentes. La observación que vimos es que estas bacterias se encuentran en lo que parece ser corales muertos, y no se encuentran más arriba donde están los corales vivos. Entonces, al parecer, esta observación meramente de lo que estamos viendo en este muestreo, no sabemos si hay una correlación con la presencia de estos microorganismos y la salud del coral. Estos microorganismos sabemos que utilizan azufre reducido para poder sobrevivir, lo utilizan como donador de electrones para producir energía, y este azufre reducido puede provenir de otros microorganismos que son sulfato reductores. Estos sulfatos reductores se encuentran también asociados en la degradación de materia orgánica, son muy importantes en el ciclo del azufre, y lo que hacen es utilizar el sulfato como último aceptor de electrones para producir este azufre reducido. De hecho, todos los organismos necesitan azufre para... Hay dos proteínas esenciales que requieren, dos aminoácidos esenciales, metionina y cisteína, que requieren azufre. Y la mayoría de organismos necesitamos este azufre. Hacemos reducción de sulfato para generar estos aminoácidos sin necesidad de obtener energía. De hecho, se necesita ATP para producir esta reacción, para biosintetizar estos aminoácidos. En cambio, estos microorganismos que vemos aquí con estos pelitos blancos utilizan este azufre reducido para producir energía. Estas condiciones son anóxicas y es decir, ausencia de oxígeno. Y ahora vamos a explorar esta zona, que son unos 1500 metros a lo largo, y estamos empezando a explorar esta zona rocosa. No habíamos encontrado sitios que tuvieran esta característica, que permitieran el desarrollo de corales, y bueno, esta mañana Brett decidió muestrar esta zona, y con esta sorpresa tan grande. Así que gracias por sintonizarnos, estar aquí, y vamos a ver qué encontramos en esta zona. Estamos meramente explorando. Por ahí había una pregunta acerca de cómo perseguir una carrera en marine biology. So I'm gonna ask people in the room how do you get into marine biology as a profession, Jacob?
I saw that question. I've been thinking about it for the past 20 minutes.
Yeah.
And I think you'll get about as many different opinions as marine biologists do ask. So you can always keep asking. If I had some general suggestions, it's that marine biology is really biology first, and that goes for most of the marine sciences. So, within the marine sciences, there are chemists, there are biologists, there are physicists, there are biochemists.
If you want me to invite her,
there are fluid mechanics. There are people in all kinds of scientific specialties, and you really have to learn the fundamentals of whatever field you want to go in, and then take it into the ocean.
We don't need
and so the only sort of three general suggestions I can give are, if you want to study biology in the ocean, get good training in biology, spend a lot of time in the ocean, and then when the time comes, try to find a team of marine biologists and make yourself useful.
Yeah.
Which can be tricky because it can be hard to find a job that pays in marine biology, especially in the early stages. But if you have a solid background in whatever kind of biology it is you want to do, whether it's ecology or molecular biology or something else, before you decide to specialize in the ocean, I think that sets you up well.
Tenemos en esta expedición expertos como Dr. Lu Chao, that's sitting next to to me, who don't have a background in marine biology, but as a part of like international collaborations and expeditions, then you can get to explore and have opportunities like this, right? Luke, what can you tell us about how to get into ocean exploration without having a background in marine biology?
Well, I think the really fabulous thing about science is that there are a huge number of opportunities that can be open to you just based on the interactions you can make. And so for myself, our group, we're trained as biochemists, as structural biologists, so it's rather basic fundamental biology, but it has tremendous applications. And for a lot of us, we're curious about questions of evolution, of symbioses, and even if we are interested in human diseases, there's a lot of relevance and things that we can learn just exploring marine ecosystems. Just a little bit of background where I'm coming from. I'm based in the United States at Massachusetts General Hospital. I'm in the Department of Molecular Biology. And so our group, like I mentioned, we're biochemists, structural biologists in a basic science department. Even though we are in a hospital, our department and our group is really fascinated by fundamental questions regarding how membranes are shaped. And these are principles that are conserved throughout evolution, from land-walking organisms, from big organisms to tiny microbes.
Thank you, Luke. Do you want to add something else, Bas, to how do you get into ocean exploration without really having a marine biology background? Now, Professor Bas.
Thanks for the question, Val. Yeah, so this is Buzz Baum. I'm a researcher, a cell biologist from the MRC LMB in Cambridge. and I'm thrilled to be here in the mission control where we're exploring this reef at 200 meters down. And yeah, as Luke said,
What do you want? I can do.
what an amazing opportunity to be here, looking at the bottom of the sea. And for me, I guess it's been a long journey, but I studied molecular biology, cell biology, genetics, all these tools by which to study life. And what's amazing is that we now know that all of life on Earth comes from one event, that every living thing on Earth is descended from one organism. And because of that, if you study yeast or flies or worms or bacteria or archaea, you can learn lessons about everything else that's alive, because everything speaks the same language, is made from the same kind of small molecules assembled into similar polymers. And so while there's all the diversity of life on Earth, which is so beautiful to see, we saw this amazing shark, all these corals, anemones, bacterial filaments, there are things that unify them. And so for years, I suppose, like many biologists, I was studying the fundamentals of things that unite all of life. But it's wonderful to be thinking now about life in the context of evolution ecology, where you start to appreciate all the differences. And so I'm learning about those. And then, of course, the Asgard Archaea, which we're in search of, the reason they're fascinating is because they maybe provide clues as to how we transition, how life on Earth transitioned 2 billion years ago from simple cells to complex cells. And as we're coming to learn, the simple cells are not so simple. We assume things about life, but actually often it breaks the rules that we think apply. And so we're learning to sort of understand the complexity, not just of individual bacterial and archaeal cells, but also how they live in communities, which are a bit like rainforests or coral reefs, but maybe in the mud. So it smells of hydrogen sulfide, it's black and sticky, but from a microbial perspective, it's as rich as a rainforest. So yeah, I'm hoping that by being here with Fraser from my lab at the MRC LMB in Cambridge, we can contribute something to the expedition by bringing our knowledge of archaea in the lab. Fraser's an expert in growing them and looking after them. We can bring that to the group. And so when we take samples from here, we can take them back to MRC LMB in Cambridge and maybe find creatures that we didn't know about before, or community organization that we didn't know about before, and apply the tools that we've learned doing molecular cell biology to questions in evolution and ecology, and really understanding more about this beautiful planet that we live on. Over to you, Luke.
Thanks, Buzz, for that really nice description of your lab's work.
And it looks like it's exposed to a lot of current. And that probably means a lot of tasty particles for these corals to eat. scaling still here? It makes me wonder whether the sulfidic water coming out of these things is really from an aquifer, like under the upper layer of the crust, or if it's just like fermenting organic matter
It's like,
down inside these mounds,
it's
which
like
are very
Greenland
porous.
in the fall.
Yeah.
Yeah. Yeah.
Yeah.
Yeah. Ian's referring again to some of our recreational diving escapades. If you go off of if you go off the coast north of Boston in the fall, you find a lot of decaying organic matter and actually some of these same macroscopic sulfur bacteria.
Current on this side's a lot stronger.
Yeah.
We're kind of sheltered from it.
Back
Around
off
the
if
other
you
way.
want. Or, yeah.
I mean, it's alright.
Okay.
We're still good. Still just try to battle and come round, but
Okay.
tell that it's, we see it.
Yep. For sure.
It's all pushing that way.
Yep.
I haven't seen any of these sharks before today.
No? Just the like smooth out, like the dogfish ones.
That's a nice looking fly trap anemone.
Ooh.
I think. Yeah.
Maybe that couple.
We're gonna start up soon, yeah.
Yeah, like
Okay.
5 minutes.
Jake, this is taking the last shit if you want to swap out of it.
Yeah, I might sign off. I'll be back later, provided we get in the water this afternoon. Thank you all for joining us.
Give her some space.
This is Jacob, ship
cooler back
substitute
here.
teacher, out.
What's up, everybody? This is Ian Hughes coming in on watch. You may have heard my ramblings in the background. Now I'm here in the foreground. But yeah, for those of you who are just joining us, we are exploring a reef off of the coast of Uruguay. Estamos explorando un arrecife 200 millas este de Uruguay, como Paloma. Yeah, there's lots of cool corals and anemones. One of our highlights, I know probably most people are pretty excited about the corals and the anemones because they're objectively cool. I agree, but one of our highlights for this particular cruise has been some of the bacteria. So we're stoked to see that kind of stuff. These sort of hairy bacteria are usually indicative of sulfide. And so typically this kind of sulfide situation is usually sort of further in the sediment. So those of you who joined us yesterday may have seen some of that black anoxic sulfidic sediment that's coming kind of underneath the surface. And so what we're looking at today are a few spots where some of that sort of anoxic sulfidic water is probably bubbling up a little bit, or at least sort of diffusing up. And but yeah, so now we're kind of just scooting around this reef to see what kind of stuff we find. I gotta say, I do not know the name of the shark that we saw. It looks kind of like a swell shark that we'd have in the US. Not huge. Unfortunately, if it's a fish or a shark or a bird, I'm not necessarily the best person about it. I don't think we know exactly the type of shark, but we can try to get back to you and let you know.
I'm gonna head out and deal with some stuff.
Yeah.
Ascend in 5 minutes.
Perfect. So those of you who are just joining us, we have a combination here of, it's sort of Cnidarian paradise. So we have a combination of true corals, what we believe to be our true corals, like these white corals that we see on the top, as well as anemones. And so the sort of more pink fellas that we have that are forming these things, these sort of groups of clusters are very likely anemones. We also have some solitary corals, and then every so often we see these hydromedusa floating by. So there's all kinds of cool cnidarians. For a question about the pH is 7.97, which is pretty standard to what we've been seeing around here. And so one of the cool things about this particular region is that we're right on the shelf. And so this is right where we go from sort of 50 to 200 meters, and then it drops down quite dramatically to well over 1,000 meters. And so what happens here in Uruguay, which is really cool, is that we have currents that are really cold from the south. This is like the Malvinas current. And then we also have currents that are really warm from the north, from Brazil. And so that northern water is warm and it's salty. And that southern water is cold and it's often pretty fresh. And so what happens in this area is that you have a mixing of this cold and fresh water and this hot and salty water. And what's really kind of interesting about this whole situation, if you're into physical oceanography, ignore it if you're not, is that basically water that is colder is denser and water that is saltier is denser. But what we have in this situation is hot, salty water. So it's warm, so it should be floating, but it's salty, so it's a little dense. And we have cold, fresh water. So it's cold, so it should sink, but it's pretty fresh, so it should float. So this is really weird because we have water that is the same density, but it differs in its temperature and its salinity. And so the profile of the water column here is very, very interesting and strange. So that's kind of cool. One of the fun things about it is we have terms in oceanography, legitimate terms in oceanography for these types of waters. And we call this sort of like isopycnal gradients, so like similar density gradients. But this hot, salty water, we call it spicy. And this cold, fresh water, we call it minty. You can probably figure out why. And so when we're looking at water that has the same density, sometimes you have minty water and sometimes you have spicy water. And those are the actual legitimate terms that we use semi-seriously and sometimes very seriously in oceanography. Yeah, we can take a zoom in and see if... Yeah, so this kind of hairy stuff, this is this sort of exciting kind of bacterial colonies. There's a few bacteria that it could be sort of, yeah, different types of bacteria, but the metabolism, the sort of morphology, the shape of these colonies is usually a sulfide oxidizing bacteria. And so this is exciting, but one of the things that we don't really know is that sometimes basically, if something just kind of dies and there's organic matter, we have these patches. But sometimes, in the cases of things like methane seeps, it's bubbling up from below, and so it could indicate some really interesting sort of geological parameters and things like that. which is part of the reason why we've been using the temperature probe to try to see if it's warm. But it looks like it is not any warmer than the ambient temperature. So it may just be that these are patches of kind of dead organic material that are just driving productivity. And this is kind of like a really, really, really mini version of a whale fall, where if you're familiar with whale falls, we have these eventually a sulfitic stage where you get all kinds of patches of this type of bacteria. And so it may be that we just have like a dead fish or a dying coral that is causing this whole kind of scene.
Just got a small vessel move for you control.
Ian, can we tell you anything about the color of these blooms?
Yeah, just a small move.
It's pretty common that they're white. It sort of depends. So like this white hairy, but they can also be like a little bit yellow fairly frequently, or sort of different tinges of shades of kind of cream, pink, a little green occasionally. But yeah, it's hard to know from sort of this macro scale alone exactly what it is. But if you do a lot more DNA sequencing and things like that, you can figure out exactly what kind of bacteria we're looking at.
Very cool.
All right, so I think we're good to head up whenever it works for y'all.
Yeah, works for us now, I think.
Sweet.
All good. Bridge, ROV, that's us off bottom.
Muchas gracias por estar aquí en esta transmisión. Ya procedemos a subir. Fue una mañana muy emocionante el encontrar estos corales, aunque no es parte de nuestra misión. Pero definitivamente queríamos explorar estas estructuras que se veían en el mapeo que se realizó anoche. Y bueno, hasta ahora no habíamos visto estas estructuras rocosas, por eso decidimos explorar un poco. Claramente hay mucho más por explorar. Simplemente queríamos hacer un avistamiento para ver qué tipo de estructuras nos encontrábamos aquí. Como Ian mencionaba, estos corales que se encuentran a 200 metros, was it 200 meters that we were? 200 metros de profundidad. Estos corales que como comentaba Ian, no tienen endosimbiontes, por lo tanto no tienen esos colores característicos que vemos en los corales, en los arrecifes de coral que se encuentran a unas profundidades más someras, a unos 30, 40 metros. Is that right, Ian? What's the depth of the corals that you like, the corals? Yeah.
Yeah, like 150 max.
Yeah. Y sí, fue una mañana de exploración, y agradecemos todas sus preguntas. Aquí en Mission Control está Claude y Mike y Alex. Y bueno, ahora procedemos a subir. What's the most exciting thing that you saw, Ian, that you can tell from this dive? What was the most exciting feature? Yeah, that you from these corals.
Just a lot of productivity. Yeah, there's just like, we've seen so much like particulates in the water. Also on the surface, there's just been so many whales and cool animals. And so it's cool to see that we see it both on the top and when you get to the bottom, there's a bunch of cool animal life as well as bacterial life too.
We saw a whale when the ROV was being deployed, and yesterday the sea lion when the ROV was being recovered. So that's been really, really exciting. Nos quedaremos hasta el 3 de diciembre, es cuando regresamos. Así que tenemos unos días más de exploración, de análisis, de muestreos. Todavía nos quedan unos dives que hacer. Y bueno, estamos muy contentos de haber encontrado estos corales. Y en un rato más tenemos pensado otro dive, y este va a ser justo para recuperar sedimentos. Entonces estén al pendientes de la próxima transmisión, que va a ser hoy más en la tarde. I don't remember exactly the time, but stay tuned for the next live stream.